Okay, we are gonna get started with chapter 14. Just some reminders for the week. Your homework is due on Wednesday, quiz on Friday. Your test next week will be Wednesday or Friday. We'll decide kind of depending on how we're doing. It's a big test because there's 5 chapters. Chapter 11, self-communication. Chapter 12, mitosis. Chapter 13, meiosis. Chapter 14, which is Mendelian genetics kind of stuff, and chapter 15, which is the chromosomal basis of inheritance. It's a lot of stuff, but it's stuff that I feel like we're getting reinforced in lab well, hopefully. So there's no other way to split it up. The next 3 chapters that we'll cover, 16, 17, and 18 are a little more intense. So I didn't want to add stuff to that test. So that's why it's broken up this way. This week in lab, you're going to be doing Mendelian genetic stuff. So we're not going to do a whole ton of Punnett squares in lecture because you're going to do a whole ton of Punnett squares in lab. Monotopic crosses, diagonal crosses, and pedigree analysis. So, this will be reinforced. We're lining up pretty well with what's going on in lab. Feel like we might get through 14 today. We'll see. Hopefully we'll get all the way through 14. Because that'll put us in a weird place for next week. Okay, questions about schedule. Okay, so chapter 14 is about Mendel and the gene idea. So we're talking a little bit about the history of how we know what we know about genetics, and how we got there. This is pretty basic stuff overall. I think that you probably, if you've taken high school biology at least, are somewhat familiar with some of these things. And we're talking about Mendel, Gregor Mendel, because he's the guy that figured out a lot of this stuff. He's what we call the father of modern genetics, and he won the Nobel Peace Prize for it. Okay, he did a lot of work with seeds. Depending on who you talk to or what source you read, he read and grew anywhere between 10,000 and 30,000 seedlings, which that's a pretty big range. But this happened in the 1800s, so maybe we just don't have really great records. So we're going to talk a little bit about the work that he did, what he figured out from his work, and what that means for us. So Mendel, as we said, was a monk. He actually joined a monastery at the age of 21. He wanted to be a teacher, but he failed the teaching exam. So he decided to become a monk instead. And he went to university in Vienna and studied physics and chemistry, but he worked with a mathematician, and he also worked with a botanist, and that kind of led to his work. So he had a good basis of science, and he went into this monastery that was really interested in plant genetics, which I think is really interesting that there's a monastery that was interested in plant genetics, but he had some agricultural training growing up, so he decided he was going to work with pea plants. So we're going to ask first, why peas? Okay, what are the benefits of peas? Why would he work with peas? Because they're delicious? No. Okay, that's the question I'll ask him Wednesday, if you like peas or not. Okay, why did he pick peas? What would be benefits of working with peas versus rats or something like that? Yeah, they have a fast generation time. Or a short generation time, however you want to think of that. Okay, doesn't take that long to make some more new pea plants. So it's easier to work with that. Why else peas? There's a lot of traits where they have 2 distinct things that you can observe. So there's a lot of different varieties. And many traits with 2 choices. So he ended up actually observing 7 different traits of pea plants. One of them, as you can see here, was flower color. He also looked at plant heights, the shape of the pod, the color of the pod, the shape of the seed, the color of the seed, the position of the flower, and then flower color. So he looked at 7 different traits of pea plants. What else would be good about studying peas? If you're hungry, you have something to eat. At the end, no. I mean, yes, but They also produce lots of offspring at one time. So you can get a lot of data pretty quickly. And there are ways that you can control the mating of peas. Yes, peas mate. I don't think we think of flowers as mating, but those parts that are inside the flower are sexual reproduction parts. They're void parts and growth parts, and fertilization has to happen. Usually that happens via pollination with a pollinator, but because we understand the anatomy of the flower, and they understood the anatomy of the flower in the mid 1800s, he could control fertilization. So he actually would take a flower and he would remove the stamen from it. I'm giving up on that. So he's removing the male part of the flower. So when he removes the stamen, there's no more pollen. That flower can't make pollen anymore, so it can't self-fertilize. So peas are self-fertilizing, so they can produce pollen to fertilize their own eggs. But when you remove the stamen and there's no pollen, then they can't self-fertilize. Control mating. You can control the mating. That's a C. So he would remove the stamen of a flower and then he would fertilize. So he would complete fertilization. So he would take here he's taking the white pollen and putting it on the purple, which is the female part. So he could take the pollen from one flower and use it to fertilize another flower, and he could keep them from self-fertilizing, so he could control what was going on. He could know what was happening or what flower was fertilizing what, so he would know the parents. He understood the parent generation. So after fertilization happens, he would just let those ovaries mature until you got pods that had seeds in them. And then plant the seeds to see what would happen. So this is the pea generation. The seeds that grow are then called the first filial generation, or the F1 generation. So he found when he took a purple flower and a white flower and he crossed them, he got all purple flowers. And it didn't matter if he used the white flower pollen and the purple flower egg, or the white flower egg and the purple flower pollen, he got purple flowers every time. And this kind of went against the theory of the day, which was the idea of blended inheritance, which was if you got one trait from one parent and one trait from another parent, you would get some kind of like intermediate, like you would take a purple flower and a white flower and you get a light purple flower. But he was able to prove that that is not what happens. If you take a purple flower and a white flower, you get a purple flower. And so he did this 10 to 30,000 times over a period of 8 years. And he came up with some theories and some laws of genetic inheritance. And he published them in 1865, and nobody cared. So nothing really came of it for a while. It took a while for his ideas to take root. That was a joke. Okay, it took a while. You gotta have a sense of humor here. So he picked peas as a model organism. He came up with some ideas, and he did this over and over and over and over. So he took this parental generation. He used what were called true breeding parents. So true breeding means they always produce offspring that look like them. So he took purple flowers that always had purple offspring, and he took white flowers that always had white offspring. As a parental generation, when he crossed them to make the F1 generation, he found they were all purple, but we're going to get this word hybrids in a minute. So he either let them self-fertilize the F1 plants, so he let these offspring either self-fertilize, meaning their own pollen fertilizes their own egg, or he cross-pollinated within the F1 generation. So he took these purple flowers, these F1 flowers, and he crossed them with more F1 flowers. Whether it's self-fertilization or cross-fertilization, it doesn't really matter because you get the same results. That's what I was looking for. See, all my. So the next generation is the F2, the second filial generation. And he found in this generation, it wasn't all the same. He got 705 purple flowered plants and 224 white flowered plants, which is an approximately 3-to-1 ratio. In genetics, we talk about ratios all the time. So it's about a 3-to-1 ratio. And what he found was this phenotype that looked like it was gone was back in the next generation. And this led him to some different ideas. Basically, he came up with the idea here of dominant versus recessive. Just this idea that there must be a gene for white flowers that is masked in these purple flowers, that those purple flowers are able to pass on to the white flowers in the next generation. You don't see it, but it's there. And he did this not just with flower color, he did seed pod, seed shape, pod color, pod shape, flower color, flower position. So he did this over and over and over and over and kept meticulous documentation of this. And he found this 3-to-1 ratio in the F2 generation pretty consistently. And so that enabled him to come up with, and we're going to talk about these. We're going to come back to this. I changed the order of the slides like 10 times. So he came up with 4 basic principles that were the model of inheritance of how genes or how traits are passed from one generation to the next. Using pea plants as a model plant, but being able to expand this model to lots of other traits also. So the first principle of the model of inheritance is that alternative versions of genes account for variations in the inherited characteristics. So alternative versions of the genes explain why there's variations in characteristics in offspring. In other words, there are dominant and recessive forms of genes. And because of that, offspring look different. So these alternative versions, in this case, he was really talking strictly about dominant and recessive. And we'll talk about the fact that we know that there's stuff that's not just simple dominance, but his experiments were figuring out the concept of simple dominance, of dominant and recessive forms. Second idea is that for each characteristic, and your book uses the term character for characteristic, so we'll be consistent. For each characteristic or character, an organism inherits two alleles of a gene. One from each parent. And this is something to us that is kind of like, yeah, we already know this. Okay, you've probably heard about this before. Oh, you got that trait from your mom, you got that trait from your dad. You know, like, you probably have heard this before, but this was big. This was a big breakthrough in our understanding of genetics in the late 1800s, early 1900s. People did not know this. And if the two alleles are different, so if you get both a dominant and a recessive gene, the dominant allele determines the appearance of the trait. the recessive allele is not visible. So if you have a trait where you have one dominant allele, like one purple allele, and one recessive allele, like one white allele, all you're gonna see is the purple allele. Again, this is talking about. I am hosting now, as I believe I was hosting before. My internet connection is unstable. Okay, we'll try this again. I'm not going to rewrite all of them. Oh, there. There we go. Okay, the words are back. Okay, so dominant is what's visible. Recessive is not what's not visible if there are different alleles present for that trait. And the fourth one is what we call the law of segregation. So we're going to get two laws out of Mendel's experiments. The first one is the law of segregation, which just says that the two alleles that you have for each trait will segregate or separate during the formation of gametes. So they're going to end up in different gametes. And we know this because we understand what process makes that happen. What process makes them separate? Meiosis. So mitosis is making exact copies, but meiosis is splitting things in half. So it's taking these two alleles and putting them in different cells, moving them to different cells. So this happens in the process of meiosis, which we just talked about last chapter. Okay, so that's happening in meiosis, and that's the law of segregation. That the two alleles that you have for a trait, or the two alleles that the P plants have for the trait, whatever, are separating during meiosis and going to different gametes. So this is showing us the chromosomal implications of this. So this is showing you, here's a chromosome. This is a pair of homologous chromosomes, showing you the different locus where the flower color gene is located. If you zoom in on that flower color gene, you can see it's just a sequence of DNA. So there's a short sequence of DNA that is associated with flower color in a pea plant. This is probably not actually the whole thing. But you can see the only difference here between the purple flower and the white flower is one base. So in the purple flower gene, it's showing you it's a cytosine, and the white flower gene is showing you it's adenine. When cytosine is present, that is the directions. These are the directions that make a specific enzyme, and that enzyme is going to help synthesize the purple pigment. If you don't have that cytosine there, you don't have the right instructions, so the enzyme can't be made, so there's no purple pigment. But if the flower has just one purple pigment gene, that's enough to make the whole flower purple. So it's still going to be all purple. But it's a change in the sequence of the DNA that determines whether or not that enzyme is present, that will determine whether or not that plant has pigment. And we'll talk more about this DNA molecular side of the way the traits are inherited in chapter 15. Okay, I need to go back to that one slide that I skipped. So before we get into what does this look like when we are doing predictions and things like that of different genetic crosses that can happen. There's some genetic terminology that I want to make sure you're familiar with. This will be helpful for you in lab this week as well, because you're going to be using these words over and over and over and over and over again. So we'll look at this cross, and we're looking at the F2 generation of the cross of the flower color cross. And you can see we have 3 purple flowers and 1 white flower. These are what we call the phenotypes. Okay, when we say phenotype, what do we mean? Yeah, what does that gene actually look like when we see the physical expression of it? So that's what you can actually see. So you can look at that flower and you can say, hey, you're purple. Do I know what kind of genes they have just because they're purple? No. But I can tell you the phenotype ratio of the offspring. So these purple flowers would have the dominant phenotype. The white flowers are the recessive phenotype. Okay, on the other side, we have the genotype. So what do we mean by genotype? It's not what you actually see, it's the type of genes they have. Okay, it's the alleles or genes that are present for a trait. So if you remember, we said that white is recessive. The white flower has 2 alleles and it has a recessive phenotype. So what 2 alleles do we know that it has to have? Does it have dominant alleles? Does it have recessive alleles? Or does it have one of each? It has to be 2 recessive alleles. So your book uses P's, but I don't like P's because it's really hard to tell the difference between a P that's capital P and a P that's a little p. So we're just going to use G's because that makes it easier to tell. Even though it has no correlation to purple or white. Okay, so this, in terms of genotype, we can look at something that is recessive and we can tell what its phenotype is and its genotype. Because if you're showing the recessive phenotype, you have to have 2 recessive alleles. So little g would be what we call homozygous recessive. Homozygous meaning the 2 alleles are the same. Homozygous recessive, both of them are recessive. In terms of the purple flowers, we actually have 2 different genotypes. So one of the purple flowers is going to have 2 dominant alleles. What would we call that? Homozygous dominant. So having the same alleles, both alleles are the same, both of them are dominant, which means the other 2 have to be what in terms of genotype? They are big G, little g. Did you ever read that Dr. Seuss book? The ABCs? Big G, little g, what begins with G? That's what I think of every time. I'm not going to recite the whole book, but these are what we call heterozygous because their alleles are not the same as each other. We also use the term hybrid for this. We would say that this plant is a hybrid for flower color because it has 1 dominant and 1 recessive allele. Thank you. There we go. So we talked about homozygous, we talked about heterozygous, we talked about genotype, we talked about phenotype, we talked about hybrid. And the other word I wanted to talk about was test cross. We talked about true breeding. Before we do that, let's talk about those. Both the homozygous dominant and the homozygous recessive ones would be what we call true breeding plants, meaning their offspring always have the same characteristic as them. So the offspring are always the same as the parents. So we can look at a plant and say, oh, we know that you are homozygous recessive because we're showing a recessive phenotype. We can't look at a plant that is purple and say, I know what your genotype is. Because a plant that is purple could be homozygous dominant big G, or it could be heterozygous big G, little g. So what we can do to figure that out, if it's a hybrid or if it's a true breeding, is to do what we call a test cross. So a test cross is to figure out do you have a plant that is heterozygous big G, little g, or homozygous dominant big G, big G? So a test cross will answer that question. I'm not going to tell you what we're going to cross it with. We're going to get back to that. So how do we figure out, how are we going to figure, well, yeah, I don't know. What we do, I'm just going to tell you, we'll do a Punnett square of it. What we can do for this to do a test cross is we're going to cross the plant that we don't know. We're going to cross it with a plant that is homozygous recessive. And if we cross it with a plant that is homozygous recessive, we can tell what the genotype of the parent is. So we're going to do some Punnett squares to figure that out. Okay, to show you how a test cross will prove what the parent looks like. So we take, to do a test cross, we take a plant that has the dominant phenotype that we don't know the genotype of, and we cross it with a homozygous recessive plant. So a Punnett square is a tool that we can use to figure things out, to make genetic predictions. So we can predict the composition of alleles of an offspring with some accuracy, or the probability of them with some accuracy. So if we have a plant, we'll start with the easier one. If we have a plant that is heterozygous, I'm sorry, not heterozygous, homozygous dominant, and we cross it with a plant, this is that is homozygous recessive. So this is how we can tell the test cross thing. So we have this plant. We know that the alleles are going to do what during meiosis? They're going to separate. So one allele is going to get this, and one gamete is going to get this, and the other gamete is going to get that. So that parent really can only pass on a homozygous, can only pass on a dominant gene. If you're homozygous dominant, you can only pass on a dominant gene. So we put those possibilities up here. This plant, which is homozygous recessive, can pass on what? Can pass on a little g or a little g. So we put that here. And then we can fill them in. Now, when you fill in a Punnett square, you always write the dominant one first. So you bring this one down and this one over. This one down, this one over. This one down, this one over, this one down, and this one over. Okay, this is a single trait Punnett square. They're not even really that fun. After a while, they're pretty easy and they aren't as much fun as we'll get to with a dihybrid cross, which is really fun. Okay, after you do a Punnett square, you're going to figure out two ratios. You're going to figure out the genotype ratio of the offspring. This is obviously a pretty easy one because they all look the same, but you always write them in this order: homozygous dominant, heterozygous compared to homozygous recessive. So in this case, how many of our offspring are homozygous dominant? Hold up your hand. 0. So I'm going to put a 0 there. How many are heterozygous? 4. How many are homozygous recessive? 0. So I have a 0 to 4 to 0 ratio, also known as a 0 to 100% to 0 ratio. You can also make a phenotype ratio. And in this case, you always do dominant first, followed by recessive. How many of these would show the dominant phenotype? How many of these would be purple flowers? What color would this one be? So this one's going to be purple. This one's going to be purple. This one's going to be purple. And this one's going to be purple. So how many of them have the dominant phenotype? 4. How many of them have the recessive phenotype? 0. So 100% of them would be purple. 100% of them are heterozygous. So if I was setting up a test cross and I didn't know if my parent was big G, or big G, little g, and all my offspring turn out to be purple, I know that my parent had to be big G, because all the offspring end up purple. Let's do what would happen if we had a heterozygous parent. Sorry, I added more last night after I posted it. Okay, so take a minute and see if you can do this. Set up the Punnett square and figure out the genotype and phenotype ratios. And then filling in your Punnett square. Okay, starting with our heterozygous parent, what can it pass on? When the alleles segregate, it's going to pass on. Some of them are going to get big G, and some of them are going to get little g. Our homozygous recessive parent can only pass on little g's. So as you fill these in, what color flower will this one have? Purple. What flower will this color flower will this one have? White. Purple. And so what is our genotype ratio? How many homozygous dominant do we have? 0. How many heterozygous offspring do we have? 2. How many homozygous recessive? 2. Phenotype ratio. How many purple do we have? How many white do we have? So if we were doing a test cross, because we didn't know the offspring, the possibility of this one, we didn't know what its genotype was, but we cross it with a homozygous recessive plant and we get 50% purple and 50% white, then we know the parent had to be heterozygous. Does that make sense? So if we cross it with a homozygous recessive, if we end up with all purple flowers, we know that the other parent is homozygous dominant. If we end up with half purple, half white, we know the other parent has to be heterozygous. So the test cross can tell us the genotype of the parent that has a dominant phenotype that we don't know the genotype of. I'll put in a couple more. You will do a lot of practice Punnett squares in lab. And again, the 4 square Punnett squares aren't super crazy. After a while, you can do them in your sleep. That's illustrating, as we've already said, Mendel's first law of genetics, which is the law of segregation. So this is just the idea. Oh, wow, I didn't spell that right. That's just illustrating the idea that during the process of meiosis, these alleles are going to separate from each other and they're going to end up in different gametes. So in this case, this parent cell has two different combinations of alleles that it can pass on, four different combinations of alleles, sorry, two, that it can pass on to its offspring because of the way that the alleles separate. The second law that Mendel discovered and is credited with is the law of independent assortment, which we've already kind of talked about when we talked about in meiosis, the fact that it creates genetic diversity. But we'll talk about how he figured this out. The law of independent assortment says that alleles on non-homologous chromosomes so on chromosomes that are not paired up, assort independently. So this allele has no effect on where this one goes because we know from meiosis that they can line up in any order. The maternal chromosomes and the paternal chromosomes can be on different sides. And so they don't affect each other. How do you think he figured this out? How would you figure out that genes don't affect each other? Yeah, if you look at multiple traits at the same time, you can see if one is always associated with another. So does a tall plant always have green pods, or can a tall plant have yellow pods? Does a short plant always have yellow pods, or can a short plant have green pods? So he did this by studying two traits at a time. So he took plants that were true breeding for both characteristics, and he crossed them as the P generation. And then he looked at the F1, and then he crossed the F1 with each other and looked at the F2 generation. So same thing he did before with one trait, but now he's looking at two traits at the same time: plant height and pod color, or seed shape and seed color. And this is where the fun really begins, and we'll have time to get through one of these at least, so that's good. So we'll look at his experiment here. This is looking at seed color and seed shape. So he was looking at yellow seeds that were round, so they're true breeding, and he's crossing them with green seeds that are wrinkly. So we're going to work through how you figure out the gametes of this, because this is where people get tripped up on dihybrids. And again, you'll practice these in lab. Okay, if this parent is true breeding for both seed color and wrinkliness, seed shape, what are the only alleles that this parent could pass on? In terms of seed color, so think of it as the two different traits. In terms of seed color, what's the only one it can pass on? If it's homozygous dominant for seed color, it can only pass on the dominant allele, which is big Y. Again, not a big fan of Ys, but. And for seed shape, if it's homozygous dominant for round shape, what is the only allele it can pass on? Big R or little r? Big R, because that's all it has. So this parent can only pass on big Y, big R. How about this parent? It's homozygous recessive for seed color. What can it pass on? Big Y or little y? Little y is the only thing it's got to pass on. And in terms of seed shape, little r, what's the only thing it can pass on? A little r. So when we put those together in an offspring in the F1 generation, we get an offspring that has big Y, big R, little r. What color seeds will it have? Which one's dominant? Yellow, so it's going to have yellow seeds. Will they be round or wrinkled? They're going to be yellow, round seeds. Why? Because it has a dominant allele for seed color and a dominant allele for seed shape. It has the recessive ones, but we don't see that in the phenotype. So it's a yellow round phenotype, 100%. These are what we call dihybrids because they're heterozygous for two traits. They have a dominant and a recessive allele for each trait. So this is where it really gets fun because our Punnett square for this isn't 4 squares, it's 16 squares. Okay, so what we really need to know, the first step of making a Punnett square is always determining what alleles can the parent pass on. Okay, so if we have a big Y, and I'm just going to write it again. Big Y, big R, little r. There are 4 combinations of alleles that parent can pass on. And there are different ways to think about it. Some people can just look at it and do it. Some people, we bring back FOIL. Remember FOIL from algebra? What does FOIL mean? First, outer, inner, last. And you can remember that. So if you can remember that for this, we'll think about it this way. We're going to, well, we got to capitalize it. That's a rule. We're going to FOIL this to figure out the possible offspring. But I actually want to use, I want to use 2 colors. Why is it? What are you doing to me? There we go. So we're going to use 2 different colors because I think that helps. But I'm not using yellow because I don't think yellow will be helpful. Is that different enough that you can tell? Yeah. Okay, so you have to think of these as 2 separate pairs. So you've got your pair over here. This is a pair of alleles for one trait, and this is a pair of alleles for the other trait. So you're going to take the first allele from each trait. So what would that be? The first allele from this one would be big Y, right? Oh gosh, no, come back. So this parent can pass on a big Y, and the first allele from this one is going to be, which one's first? Big R or little r? Big R. So it can pass on big Y, big R. That colors did not work, really? Let's try. Okay, how about the outer alleles? So we did first. Now we need to do outer. What is outer? Big Y, little r. Inner, little y, big R. And last of the pair is little y, little r. If that helps you, that is great. If you don't need that, that's also okay. As long as you can figure out what are the possible combinations of alleles that they can pick, that a parent can pass on, it doesn't matter because you're going to take these 4 alleles and you're going to put them up here. So big Y, big R, then big Y, little r, then little y, big R, then little y, little r. And we have to do the same thing down the side. I did not leave myself enough room. Because both parents are the same, they both have the same possibilities of alleles that they can pass on to their gametes. So big Y, big R, big Y, little r, little y, big R, little y, little r. So once you figured out the gametes, it's a matter of filling it in. And there are some rules about how you fill it in. Okay, when you fill it in, so you're going to bring down and over. In this case, it doesn't matter too much because you've got 2 big Y's. So you always keep the Y's together and you always keep the R's together. The next one, again, you're going to keep the Y's together. Y from here, Y from here. And you're always going to write the dominant one first in the pair. So in this case, this offspring gets a big R from here and a little r from here. I'm going to write the big R first. Always. So I keep the order of the traits the same, but within the pair, I always write the dominant one first. So here, I've got a big Y from this parent, a little y from this parent. and a big R from this parent, and a big R from that parent. I've got a big Y from this parent, a little y from this parent, a big R from this parent, and a little r from that parent. So I'm keeping the alleles for the same trait together. I'm always writing the dominant one first. So go ahead and fill in the rest of it. As I'm filling it in, you can also fill in the phenotypes. So you can write whether it's yellow round, yellow wrinkle, green round, or green wrinkle based on what the genes are. Okay, so that's what it should look like once you filled it in. We're gonna count real quick. We don't ever bother with a genotype ratio with this because it's just a mess. Okay, to try to figure out all the possible genotypes would just be too painful. So we don't do that. What we really care about in this case is the phenotype ratio. So for phenotype ratio, we're looking at the one that is dominant and dominant compared to one that is dominant and recessive compared to one that is recessive and dominant compared to one that is recessive. Oops, see, I did it backwards. Recessive and recessive. So what I do when I'm doing this, and we may run out of time, but as I go through and I count and I say this one's going to be yellow and round. So I just write yellow round. This one's going to be yellow and round. This one's going to be yellow and round. This one's going to be yellow and round. This one will be yellow and round. This one's going to be yellow but wrinkled. This one's going to be yellow and round. This one's going to be yellow and wrinkled. This one's going to be yellow, yellow and round. This one's going to be yellow and round. This one's going to be green and round. This one's going to be green and round. This one's going to be yellow and round. This one's going to be yellow and wrinkled. This one's going to be green and round, and this one is going to be green and wrinkled. So then I just cross them out as I'm counting. I say 1, 2, 3, 4, 5, 6, 7, 8, 9. I have 9 yellow round ones. How many yellow wrinkled do I have? I mean, yeah, yellow wrinkled. I should have written it that way. Sorry. I have yellow wrinkled, yellow wrinkled, yellow wrinkled. So I have 3. How many, we're going to say green round, do I have? 1, 2, 3. And how many green wrinkled do I have? 1. A 9-3-3-1 ratio. It's classic. You'll do it in lab.